Okay, so our last lecture will be on the nervous system. So we're going to discuss the nervous system just in general, how it's organized, and then of course we will talk about metabolism, blood-brain barriers, and things like that. So first off, this is just a very simplistic drawing of the cells of the nervous system. And so of course you see here that we have our neuron, and the neuron is really what transmits signals. And so within the neuron, you have the cell body or the soma, and the cell body have long axons and dendrites. And then the dendrites are going to receive information from the axons of other neurons. So here you have an axon, and here you have a dendrite, and the dendrite receives information. Um, and so the synapse is the axon and dendrite connection. Now, neurons can integrate information from multiple sources. And they have one axon, but this one axon can branch extensively and distribute to multiple targets. And so this ability for a neuron to have or distribute information to multiple targets is simply called divergence. And this, of course, is through its branching. And the signals are going to, or across a synapse, are going to require the release of neurotransmitters. The other thing to think about is neurons are terminally differentiated. And what that means is that they are only going to be a neuron. They cannot change. They are at their final state. And so their actions, their place in the body are set. So signaling across that synapse is going to be by the release of neurotransmitters. Okay, and so let's just talk a little bit about the different cell types. We have our supporting cells. So neuron, of course, is the first cell type. And then we have our supporting cells. And so which are called neuroglial cells. So in this, we have our astrocytes. And these are going to provide support for neurons. And this is both physical as well as nutritional. So nutrients. And then we have our oligodendrites. And these are going to provide a myelin sheath that surrounds the axon. And this is going to be a sort of insulation for neurons. And we have our Schwann cells. And these are going to be supporting cells of the peripheral nervous system. And so Schwann cells are going to provide a myelin sheath, but they are limited in only that they can myelinate only one axon. Okay, and so Schwann cells are also going to have the ability to pick up any sort of debris, and it has an ability for peripheral axons to regenerate if they're damaged. Now we also have our microglial cells, and micro means small, so they are the smallest glial cell, and they are very similar to macrophages, so like our Kupffer cells in our liver. So they are going to be there to phagocytize cellular debris, to maintain homeostasis, as well as to have an impact on protection from invading microorganisms. Now we also have epididymal cells. These are going to be ciliated cells that line the cavities. And this is going to be the cavities of your ventricles and of your CNS as well as the spinal cord. Now they're going to secrete the cerebrospinal fluid into the ventricular system, and the cilia allow for the circulation of the cerebrospinal fluid. Now, cerebrospinal fluid, of course, has a function, and it's going to be a shock absorber for mechanical stress. It's also going to remove metabolic waste and can act as um, stem cells and regenerate neurons. Okay, so now your brain has what everyone knows as a blood-brain barrier. And so one thing to keep in mind is to compare this to what's happening in other organs. So in most organs, you're going to have passage between capillaries that will be able to pass things into the interstitial fluid. So this is happening in most cells. Now, of course, we talked about your leakiness of your liver, so that's a unique site. But the brain has what's called a transcapillary movement of substrates, um, and this movement, the transcapillary movement, is going to be restricted. And so this is called the blood-brain barrier. So endothelial cells are going to form the inner lining. So you're going to have your endothelial cells, and in your blood-brain barrier, they're going to be an inner lining. They're going to be joined by tight junctions. Um, and they don't permit polar molecules from the blood into the interstitial fluid that is bathing the neurons. They also lack mechanisms for transendothelial transports. So they have no fenestrations, no transpinocytosis, etc. Now, for the endothelial cells, their purpose is to protect the brain. So they're protecting them by influencing what enters. So they do have drug metabolizing enzymes, similar to liver. They are going to actively pump hydrophobic molecules that diffuse into the endothelial cells back into the blood. Um, and they're going to do this with their phosphoglycoproteins, which act as ATP dependent efflux pumps. So there are some things that can cross. So what can cross? We have our lipophilic substances. our low-density lipoproteins, for example. We have water, oxygen, of course, carbon dioxide, and also molecules required for brain function. So all of our essentials, if they say essential, it has to be able to get through. So our essential amino acids, essential fatty acids. But these are going to require specific transport. Also our fuels. We know that the brain can use ketone bodies. So ketones have to get through. Glucose, sugar has to get through, etc. So what we know that the brain needs metabolically has to be able to enter the blood-brain barrier. So the blood-brain barrier is going to be a continuous and have a continuous collagen-containing basement membrane that's going to surround your that's going to surround the capillary. So we have our basement membrane, and this is going to be a collagen-containing. And you're also going to be surrounded by foot processes of your astrocytes. And you're going to have several layers of cells that then are going to help to protect the brain from things that are actually in the blood. So here's another astrocyte. And here you have a neuron. And within the basement membrane, you have your pericytes. So compounds have to pass through to get in. Your compounds must pass through several layers. And they're going to have to pass through a luminal membrane. They're going to have to pass through the enzymatic barrier of your actual endothelial cells that are actively pumping things back out, egress. You have an albuminal membrane. You have a basement membrane. And you have other barriers formed by astrocytes. Now we'll talk a little bit about neurotransmitters. So there are two basic categories of neurotransmitters. You have small nitrogen-containing molecules, and you have neuropeptides. So I have listed different nitrogen-containing molecules, and our peptides are synthesized in the CNS. And some are going to target the CNS, such as your endorphins. And others are going to be released. And some examples of this are growth hormone and thyroid stimulating hormone. So in general, each neuron is going to synthesize and transmit transmitters it uses. A single neuron can also secrete both of these types of transmitters. They can secrete the small nitrogen-containing molecules, and also they can secrete your neuropeptides. So with respect to our small nitrogen-containing transmitters, most are going to be synthesized from amino acids. Or intermediates of glycolysis and TCA cycle. The synthesis is going to take place in the cytoplasm of the presynaptic terminal. And once synthesized, the neurotransmitters are going to be transported to storage vesicles. And the transport is going to be by an ATP-dependent pump. And this is always going to be linked to a proton gradient. So similar to all of the other ones, have to create a gradient. You're going to have one pump that does the pumping in and another pump that's actually creating the gradient. So that's going to be the same. Now, release of the neurotransmitters from the storage vesicles is going to be triggered by nerve impulses. And then the nerve impulses are going to depolarize the postsynaptic membrane and cause an influx of calcium via calcium channels. Now, the calcium influx is going to, similar to what happened in other cases, kind of cause a fusion of vesicles with the synaptic membrane, and then you get release of the neurotransmitter into the synaptic cleft. And then the neurotransmitter, of course, is going to bind to the receptor on the postsynaptic membrane, completing the transmission. The termination of the neurotransmitter is going to occur through reuptake into the presynaptic terminal or uptake into glial cells. So remember, those glial cells are like macrophages, or it can be simply diffusion from the synapse. Or it's going to be through some sort of inactivation by an enzyme. So enzyme inactivation is going to occur in postsynaptic terminals in the presynaptic terminal or an adjacent astrocyte microglial cell. It can also occur in endothelial cells and brain capillaries. So basically, in the cytoplasm of all of these cell types, you're going to have enzymes capable of inactivating neurotransmitters. And that's part of the reason why you have to have your neurotransmitters stored in a vesicle, or else those enzymes will inactivate it. Now, this is a diagram showing the synthesis of the catecholamines. All three are going to use a common pathway, and they're using tyrosine as their precursor. So here's tyrosine up here. The rate-limiting step is the hydroxylation of tyrosine ring by the tyrosine hydroxylase. Give it enough run. And this uses tetrahydrobiopterin, as we know. And then the second step requires pyridoxal phosphate. And then you're going to get, of course, dopamine. And then dopamine beta hydroxylase is going to catalyze the reaction from dopamine to norepinephrine, and this is going to use vitamin C. Vitamin C is going to be an electron donor, and copper is required for the electron transmitter. And then remember, we can also have SAM generate epinephrine. So next up is SAM. So we have a lot of vitamins involved in all of this. We have our PLP, vitamin B6. We have our vitamin C. We have our vitamin B12 and folate to generate SAM. So there's a lot of vitamins involved in making these catecholamines. So all of those are going to be involved. Now, importantly is the storage and release of catecholamines, and it helps to understand how these transmitters are released just in general. So we have very low levels of the catecholamines are actually in the cytosol. And these would actually get degraded. And that's via those enzymes. And so you're going to want to move these catecholamines after you make them and move them quickly into storage vessels. And you're going to have, for example, for these catecholamines, you have the formation of dopamine in the cytosol. And then your dopamine is going to be transported into the vesicles by your VMAT, vesicle monoamine transporter 2. And then you can see this is in exchange for our hydrogen. And so associated with this, of course, is going to be a ATPase pump. So we have our V-ATPase pump of vesicular. So the V stands for vesicular, if you remember from Biochem 1. And so the vesicular V-ATPase pump is going to pump protons into the vesicle. And the protons are going to be exchanged for your charged catecholamines. In this case, we have, of course, dopamine. Now, norepinephrine hydroxylation is actually going to occur in the vesicles. The vesicles have a dual role. So, of course, vesicles are there to store the neurotransmitters, and they're also going to be part of the process of neurotransmitter release via exocytosis following an action potential and calcium influx. Which you guys probably already should have figured that out. So finally, we have inactivation of our catecholamines. So we're always going to have termination, which can occur via reuptake into the presynaptic terminal and diffusion away from the synapse. The enzymes that are going to be able to degrade your catecholamine neurotransmitters are going to be present at the sites, including in your glial cells and endothelial cells. Now, the two major reactions are going to be catalyzed by your monoamine oxidase and your catechol O methyltransferase. So we have our MAO, which is our monoamine oxidase. And we have our COMT, which is our catechol methyltransferase. Now, MAO is going to be present in the outer mitochondria membrane. I should maybe separate these a little bit. And it's going to oxidize carbon containing the amino group and releases an ammonium ion. And so here's the MAO. And then MAO can degrade catecholamines not protected in storage vesicles in the presynaptic terminal as well. So that's one of the reasons why you want to once you make your catecholamines, you get them inside the vesicle or else they get degraded. You're going to have two isoforms of MAO. MAO A prefers norepinephrine and serotonin. And then you're going to have B, which has a wide range, so it has a wide spectrum of targets. So A is more specific, whereas B is going to be able to break down more of the catecholamines. So again, your COMT is going to be found in many cells, including red blood cells. And it works on a broad spectrum of your catecholamines and works on those that diffuse away from the synaptus. The transfer is going to transfer a methyl group from SAM. And so this is going to use SAM to add a methyl group. It's going to be dependent, therefore, of course, on SAM, which is dependent on beta-12 and folate. The actions of these two enzymes can occur in any direction. So you can start, for example, here with your Mayo, or you can have your COMT, and then you can have further reactions carried out by Mayo here, and then further reactions carried out by COMT down here. And so it really doesn't matter which enzyme works on the catecholamines first. They're both going to participate in the degradation of the catecholamines. Now, just a little bit about serotonin. Serotonin metabolism is similar to norepinephrine. So you're going to start again with tryptophan. So again, we have up here tryptophan. We have our tetrahydrobiopterin. Nothing's changed there. You then get your 5-hydroxytryptophan, and then you're going to have decarboxylation by your dopa decarboxylase, and that will lead to serotonin. And serotonin can be inactivated by your MAO, A, so this is the type A, not the type B. And then you can also get melatonin produced in the pineal gland in response to light dark cycles from serotonin. Now, there's also metabolism of histamine. So histamine is produced by mast cells and some neurons. And that's the allergy response. They're going to be synthesized from histidine in a single step that requires the histidine decarboxylase and pyridoxal phosphate. Then you get histamine. Once released from neurons, this can activate both pre- and postsynaptic receptors and is not recycled. It can also be inactivated by SAM and MAO-B in the brain. In peripheral tissues, it'll be inactivated by a diamine oxidase. So there's two mechanisms. So once in peripheral tissues, diamine oxidase, or in the brain, you first have the histamine methyltransferase, and then you have it worked on by MAO-B. Now, acetylcholine is going to be synthesized from CoA and choline. Oops, Co. Let me just do that. Sorry. And this will generate acetylcholine, and acetylcholine acetyltransferase, and it's going to be synthesized in the presynaptic terminal. It's going to be stored in vesicles. And it's going to be released via calcium-mediated exocytosis. It's going to be inactivated by acetylcholine esterase. And is going to form basically a covalent bond with the acetyl group. Acetylcholine esterase is a target of drugs or toxins such as sarin. We also have synthesis of glutamate. So glutamate is an excitatory neurotransmitter. And this is going to lead to depolarization of neurons. It's going to be synthesized from glucose in neurons, and it does not cross the blood-brain barrier. It's going to also be synthesized from alpha-ketoglutarate via transamination or glutamate dehydrogenase. So there are several ways you can get to glutamate. So here's glutamate, and so you can do this via glutaminase, where you're taking glutamine and releasing nitrogen, or you can use alpha-ketoglutarate and glutamate dehydrogenase, or you can use alpha-ketoglutarate and use a transamination reaction with PLP. The ammonium pool is going to be provided by the degradation of amino acids or ammonia diffusing across the blood-brain barrier. Now, you can also have the major inhibitory neurotransmitter in the central nervous system is going to be generated through the pathway that includes glutamate, so that's GABA. Oh, I do have a separate page for that. Let's go to the separate page. So again, we have GABA, gamma aminobutyric acid, and this is an inhibitory. So this one has many roles in neurologic and psychiatric disorders. So if its function is altered, it has been shown to be have a pathophysiological presence in such disorders. It's going to be synthesized from glutamate. So here we have glutamate. And you're going to get decarboxylation, so you release a carbon dioxide, and that is through GAD. which is glutamic acid decarboxylase. And GABA is going to be recycled by what's called the GABA shunt, and that conserves both glutamate and GABA. So you can see here that you have GABA, and through a transamination reaction, you can go back to glutamate. And then glutamate, this transamination reaction will also can form succinate as an intermediate, which can go back to TCA cycles, or be regenerated in the GABA shunt back to glutamate, and then you can get GABA made from glutamate. Now, other neurotransmitters are going to be aspartate. This is going to be excitatory. It's going to have fewer pathways than glutamate does, and it's synthesized from TCA cycle intermediates oxaloacetate. Transamination reaction that we've talked about since Biochem 1. Glycine is an inhibitory, and this is especially important in the spinal cord. So it's going to have more of an influence in the spinal cord than GABA. This is going to be synthesized de novo in the nerve terminal, and is synthesized from serine using serine hydroxymethyltransferase. As we talked about previously in Biochem 2. And this, of course, is going to use folic acid. We also have nitric oxide. So nitric oxide is a messenger in many responses. So NO is going to have an impact on vasodilation. Neurotransmission, as well as the immune system. And it's going to be synthesized from arginine via the nitric oxide synthase. And so you're going to have 3 different forms of nitric oxide synthase. So in your neurons, you're going to have nitric oxide synthase 1. You're going to have macrophage have nitric oxide synthase 2, so that's in your immune system. And then you're going to have nitric oxide synthase 3 in your endothelial cells. So what nitric oxide can do is it can lead to an increase in cyclic GMP via guanylate cyclase. So there's guanylate cyclase, and you can go from GTP to cyclic GMP. And in smooth muscles, cyclic GMP is going to activate protein kinases that help to relax the muscle and dilate vesicles. So it leads to relaxation. Nitric oxide can also cross membrane, so it can affect neighboring cells. So here's nitric oxide going across membranes and affecting neighboring cells. It is also referred to as a retrograde messenger, which can influence neurotransmitters released from the presynaptic terminals. So it's a retrograde. So it is released from the postsynaptic neuron. And then it goes to the presynaptic neuron, where it influences neurotransmitter release. And so therefore it's referred to as a retrograde messenger. So we also have our neuropeptides. Just wanted to remind that they are small peptides, and they can be autocrine in nature, targeting the central nervous system, or they can be released into the circulation system, such as with growth hormone and thyroid stimulating hormone. Now, some things that we have to discuss are some different encephalopathies and neuropathies. So we have hypoglycemic encephalopathy, which is a pathophysiologic mechanism of neuronal cell death. So basically the mechanism is where you have glutamate, excitotoxicity, and this is where energy reserves are depleted and uptake pumps fail to collect glutamate. And then you have glutamate is going to build up in synaptic clefts, leading to overstimulation of postsynaptic glutamate receptors. And this is a prolonged opening of the receptor, and lethal amounts of calcium are going to enter the neuron and activate cytotoxic intracellular pathways. There is also hypoxic encephalopathy, which can result from insufficient oxygen. This can occur at high altitudes. It can also be when you have iron deficient anemia, severe anemia, um, or cyanide poisoning. And this is going to result in diminished neurotransmitter synthesis. You're going to inhibit pyruvate dehydrogenase, so that's going to diminish acetylcholine synthesis. Acetylcholine synthesis is very sensitive to hypoxia. You're going to have diminished GABA and glutamate. Due to a elevated NADH levels, which is going to inhibit TCA cycle enzymes. And the NADH levels are backing up because oxygen is unable to accept the electrons. Electron transport chain. Also, catecholamines will decrease since hydroxylase reactions require oxygen. Now, there's also lipids. So the brain is very unique. Your cell membrane at the presynaptic terminal is going to be rapidly changing because vesicles are going to fuse and release their contents. So at the presynaptic, you're going to have the cell membrane rapidly changing. And this is due to the fusion of the vesicles. So the membranes are going to be lost. As endocytic vesicles. And on the postsynaptic terminal, the membrane contains receptors for the neurotransmitters and signaling components. The blood-brain barrier is going to restrict entry of non-essential fatty acids. And therefore the brain is constantly synthesizing lipids. So it's going to have to form fatty acids, glycosphingolipids, phospholipids, all of those. The signaling, neuronal signaling also requires that non-neuronal glial cells synthesize myelin, and myelin is lipid rich. And so that also takes a lot of fats. So since the blood-brain barrier is going to inhibit non-essential fatty acids and lipids into the CNS, they're going to make their own. Essential fatty acids can enter, remember that. So the essential fatty acids will have a transporter to get in. Another thing, myelin is going to be very long chain fatty acids are important for myelin formation. So these are very long chain fatty acid. And so, of course, or peroxisomes are going to be important in your brain to break down those very long chain fatty acids as well. So the elongation of your long chain fatty acids require 4 enzymes. And you have your elongation of very long chain fatty acid. Oops. And this is called ELOVL. You have your 3-ketoacyl-CoA. Reductase. You also have your 3-hydroxyacyl dehydratase. And you have your trans 2,3-enol reductase. So your oxidation and turnover of fatty acids in the brain occur as previously described in Biochem 1 and at the beginning of Biochem 2. Your peroxisomes are going to be involved because of the very long chain fatty acids. So any defects such as refusum disease. Let me see. Very little space here. And so diseases are going to have a severe effect on brain cells, and also the ability to metabolize your branched chain fatty acids and your very long chain fatty acids. So in addition, impairment of lipid degradation and the lysosomes in the brain become engorged, and you get neurologic dysfunction. All right, so we have N-acetylaspartate in myelin synthesis. So NAA is so here we have our formation of NAA, and if you NAA is going to be the reservoir for acetate or acetyl-CoA. This is going to be synthesized in neurons and metabolized by other cells. So reduction of your N-acetylaspartate can indicate brain injury or tissue damage. And this is coincidental or occurs alongside a decrease of ATP. And the reason it parallels ATP decrease is because NAA is a reservoir for acetyl-CoA. So this is going to be synthesized in neurons by aspartate NAT, which is enzyme here. And it's going to be that aspartate NAT is acetyl-CoAl aspartate and acetyltransferase, so it's a transamination reaction. The N-acetyl aspartate is going to be metabolized mostly by your oligodendrites. And you're going to have it metabolized by an enzyme, ASPA, or aspartoacylase D-cetylase. And then you're going to make acetate. Now, one of the last things we're going to talk about is myelin synthesis. So myelin you can think of it as a multi-layered lipid and protein structure. And this is going to be formed in the plasma membrane of glial cells. So in your peripheral nervous system, it's your Schwann cell that's responsible for myelinating the axon of one nerve, and it does this by wrapping itself around the axon multiple times. So the Schwann cell is basically forming the multi-layered sheath of membrane that surrounds the axon. In the central nervous system, it's the oligodendrite responsible for myelination. And then oligodendrite can myelinate up to 40 axons. So it can wrap around multiple, within processes, multiple axons, multiple times. So in the peripheral nervous system, your Schwann cell can only do one. In your central nervous system, the oligodendrite can myelinate numerous axons. Okay, so let's talk a little bit of myelin structural proteins. We have, let me put a little thingy here. Okay, so we have in the central nervous system, we have proteolipid protein. And this is going to be hydrophobic, and it's going to form large aggregates in an aqueous environment. It's resistant to proteolysis, and it promotes the formation and stabilization of the myelin structure. We also have the myelin basic protein. And this is going to be soluble in aqueous environment. This is located on the cytoplasm face of myelin membranes. And it is known that antibodies that target myelin basic protein elicit experimental allergic encephalomyelitis. All right, so basically you have your myelin basic protein, you have proteolipid protein, you have a lipid bilayer, and you have your extracellular spaces that have the aqueous environment. And so your proteolipid protein will span the membrane, and the hydrophobic regions, of course, will be in the membrane, and the hydrophilic regions will be interacting either with the extracellular space or with the intracellular cytoplasm. And so those are these little squiggly lines here. And then you have your myelin basic protein, which is always going to be intracellular, but it's bound to the membrane facing the cytosolic side of your myelin. Now, in the peripheral nervous system, the major myelin protein is called PO. And it's a glycoprotein that it counts for about 50% of the peripheral nervous system myelin protein content. And it has a structural role in maintaining the myelin structure, similar to the proteolipid protein does in the CNS. You have major basic proteins are also found in the peripheral nervous system, or sorry, myelin binding proteins. Major basic, thinking of immune system. Myelin binding proteins are going to be found in the major one is designated protein 2. And so that's basically the peripheral nervous system. And that is the end.